Would everybody like to take their seats? We're just about to start. Okay, good afternoon, ladies and gentlemen. We're just about to start the next session. So the next speaker will be Brian Bigger. So Professor Bigger is Professor of Cell and Gene Therapy at the University of Manchester, and he trained applied biology at Bath University and holds a PhD in gene therapy from Imperial College London. So over to you, Brian.
Thanks, Bob. I know how long that bio is, so it was good that you stopped when you did, because you would have been here all day. Great. Could I have some slides, please, do you think? So I'm going to talk to you today about stem cell gene therapy in the 21st century, or something like that, I think. If we could get the slides up, then we'll be able to. You don't have them. Oh, transition. Excellent. So I'm going to tell you about transition. No, I'm not going to tell you about transition. Do you have the Brian Bigger slide set up there? Okay, I think we might be missing the slides.
Bear with us just 1 second.
I promise I did drop them off earlier, but...
There you go.
Anyway, oh no, there we are. We got them up. Lovely. So, where were we? Right, we were going to go on to the next slide. So you all know what MPS diseases are, hopefully. So I'm not really going to tell you anything you don't know already, except that I'm going to talk quite a lot about GAGs, glycosaminoglycans, these long-chain sugars that build up in cells. And why do these GAGs build up in cells? Well, they build up because you're missing a lysosomal enzyme. And in MPS II, this is called IDS, this thing here. In MPS IIIA, it's called SGSH. Snappy title, I know. And in MPS IIIB, it's called NAGLU. And depending on which MPS subtype you've got, you're going to have a buildup of different kinds of GAGs. So we've got heparan sulfate, we've got dermatan sulfate, and we've got keratin sulfate as well. And they build up within your lysosomes. Your lysosomes help to recycle these long-chain sugars. And these long-chain sugars have lots of different kinds of functions. They help cells to signal. They help to talk to each other, that is. And in the case of dermatan sulfate and keratin sulfate, they help proper formation of bones and joints, and they help proper heart function. So interestingly, when you start looking at the different diseases, you'll notice that heparan sulfate storage diseases like MPS IIIA, B, C, and D all seem to have quite a significant involvement of the brain. And they do have some involvement of the body as well. And there's a little bit of chondroitin sulfate and dermatan sulfate buildup in some of these diseases too. Dermatan sulfate and keratin sulfate storage diseases tend to build up to have more issues with bones, joints, and the heart. And then MPS 1, 2, and 7 have both. And that probably reflects the fact that they have both heparan sulfate and dermatan sulfate storage. So, we can go back. So enzyme replacement therapies are available for any of the MPSs with somatic disease. That means the disease from the neck down, basically. So again, we're going to go very quickly through enzyme cross-correction. And the reason why I'm doing this is because when I come to the biochemical slides, you actually have a clue what I'm talking about. So if you don't want to see this, just shout skip. Okay. So lysosomal enzymes are made in the cell. They're tagged with this mannose-6-phosphate, which is like an address signal for the lysosome. It tells the enzyme to go to these lysosomes, which is where they're active. And they break down these long-chain sugars, these GAGs, okay? But some of the enzyme doesn't get there, okay? So it's not a perfect process. Some of it just goes out of the cell, and this is what we call secretion. And other cells have these receptors, these mannose-6-phosphate receptors on the cell surface, which bind the address tag, the mannose-6-phosphate, and they're able to take them up. And they can take up enzyme and send it to lysosomes. And this is the principle by which enzyme replacement therapy works. So if you deliver enzyme in the blood, it can therefore be taken up by most cells, with the exception of MPS 3C. So when we look at treatments for lysosomal diseases, the one I'm really going to talk to you about today is based on hematopoietic stem cell transplantation, which is a very swanky way of saying blood or bone marrow transplant. So the hematopoietic cells are the blood cells, and we want the stem cells, which are the cells which go on to make all of the different blood types, okay? The blood cell types. And so if you can deliver the enzyme through the blood cells, then this is a great way of getting a whole load of enzyme into your bloodstream, because these cells can release the enzyme, and it can be taken up by other cells and cross-correct those cells, and you can reduce the lysosomal size. It's also actually an extremely effective treatment in MPS I hurler. It's not perfect, it's not fantastic for bones and joints, and it's not always great for the brain disease, but it does have an impact on brain disease, quite a significant impact, especially if it's given early enough. And the reason why is because we have a type of cell from the blood called the monocyte, which traffics from the blood into the brain, where they engraft and they're able to release enzyme beyond the blood-brain barrier. And the blood-brain barrier essentially acts as like a sieving system for your blood. So you've got a whole load of stuff in your blood which would cause your brain cells to stop functioning properly. So it's really important that the brain has got like a very special media that it exists in, which is called the cerebrospinal fluid. And it's very different composition to blood. So we don't have all of the the red coloring that we have in blood, the hemoglobin, the iron, and so on. None of that stuff is there in the CSF. And that's all filtered out. So the blood-brain barrier acts as this kind of filtering system, and it filters out enzymes as well, unfortunately, for us. So whilst this is very effective for some LSDs, particularly MPS I hurler, it's not for others. And the downsides really with this treatment are that you've got to treat very early for an effect. So you're typically looking at less than 2 years old for a really good effect. There's a mortality risk with the procedure, so you have to undergo chemotherapy. It's not pleasant. And there's limited correction of bones and joints. And depending on what age you get in at, there can be limited correction in the brain as well. So why doesn't this always work? Well, we think that probably most of the problem is that many of the lysosomal enzymes are just not secreted. That's not pushed out of these cells very effectively. So whilst in MPS I hurler, the IDUA enzyme can be pushed out quite effectively from cells, other enzymes like SGSH or IDS or NAGLU just don't appear to be pushed out as well. And so here's another depiction of stem cell transplant. So this is Bart. Bart's about to have a transplant. And he's going to get his pre-transplant conditioning. So this is chemotherapy and immunosuppression. He'll then have a cell transplant, and he's got lots of different things which will affect how well those cells actually engraft. So depending on where the cells come from, depending on how well matched they are to him, so they're going to come from another individual. So the more closely related they are to him, actually the better the outcome might be in terms of engraftment. But as you'll see later, that's not necessarily exactly what you want to achieve. And ultimately, you want to achieve engraftment. But it's not just engraftment that's important. You also need these cells to produce a good level of enzyme, and not just an effect on the brain, but an effect on the bones and joints. And we know that bones, joints, bony material is actually really difficult to get enzyme into, as well as the brain. You can also take rejection in this procedure because you can be retransplanted. So it's not a complete one-way street. So why is it different in different diseases? Well, here we've got an individual who's about to receive a bone marrow transplant for MPS I Hurler. They've got very high levels of tissue GAGs, as shown by red. They get a transplant from, let's say they get a transplant from a related donor. So this would be somebody who would have an intermediate level of enzyme that have probably about 50% of normal levels of enzyme. They would be a heterozygote, i.e. a carrier. And interestingly, what we've shown is that when you take an unrelated donor, one who has normal levels of enzyme versus somebody who's a carrier who only has half the normal levels of enzyme, then actually patients who've received unrelated transplant do better. They have lower GAG levels because they have higher enzyme levels after the transplant. And you'll notice that both of these do better than an enzyme replacement therapy. So in terms of reducing GAGs, bone marrow transplant from an unrelated donor is the best outcome. And this is because they've got 2 gene copies of the missing gene, IDUA in this case, versus 1 gene copy. So what happens in MPS III, an intractable disease, or MPS II for that matter? Here's no enzyme. We've got the red GAGs. We do a transplant with medium enzyme, and we just haven't quite got a very good bone and joint disease, and we definitely haven't got a very good brain correction. And even when we go to an unrelated donor, we're still not high enough. And it's because there's not enough enzyme coming out of these cells. And interestingly, we can look at that in a different way if we look at mortality data from patients who've done transplants. And this is actually quite old data now. This is from 2008, so mortality is better now than it used to be. But this is a large number of patients who'd received transplants. And you'll notice that the 1-year survival across MPS I, MPS IIIA, or metachromatic leukodystrophy, which is another lysosomal disease, they're actually quite similar. But the 5-year survival rate is very, very different. And that's because the MPS I Hurlers are getting a much better correction than any of the other diseases. And MLD and MPS IIIA do not do well on transplant. So how do we make it better? Well, the point of this talk is about stem cell gene therapy, and the whole idea of this is to take your own blood cells and to overexpress the missing enzyme. And of course, we're still relying on these monocyte cells trafficking to the brain to produce enzyme there as well. So we would expect to have much higher levels produced in the peripheral organs down here, and we would probably have lower levels in the brain because we've got fewer cells in grafting there. On the plus side, the enzyme overexpression means that you have a more effective therapy in the brain. And even for MPS I, that would be true as well. They're your own blood cells, so you don't have immune responses against them, unlike an unrelated transplant. And you can get permanent correction because the gene therapy vector that we use integrates into your genome, and it's permanently expressed there. We know from metachromatic leukodystrophy that this has the potential to be transformative. So in patients who've received transplants, you can see here on the baseline, these are patients in gray who have not had treatment. They do really badly on their gross motor function score. So this is their ability to walk, their ability to move their arms and legs and so on, as opposed to fine motor skills, which would be things like closing your finger like that. And some of these patients who are treated are running, and they're really very good indeed. 1 or 2 have not done so well. This patient, for example, actually had a graft failure here. So it's not all perfect, but it has been really very impressive in that disease. Again, they had to be treated very early, and it still required chemotherapy. So first I'll tell you about the work that we've done in MPS IIIA, and I'm not aware of anyone else who's done work in IIIA in this sector, in stem cell gene therapy. And basically, we took the same vector that was used in that clinical trial in Milan. That was all taken on by GlaxoSmithKline, and they ran that clinical trial with the San Rafael Institute in Milan. And we took their original vector and we made it more specific for the monocyte cells that traffic into the brain. So we used the CD11B promoter here. And this promoter drives gene expression of SGSH, which is the missing enzyme, and it drives expression in monocytes. So we get enzyme produced in the cells, which are going to traffic into the brain and release enzyme. So that's really where we want it to be. So when we do this in MPS IIIA mice, we transduce MPS IIIA cells, stem cells, we transplant them back into mice, and we look in the brain. We can check to see that these cells are engrafting in the brain. So the green squiggles are the cells engrafting in the brain. They form these really cool wiggly structures in the brain, like that, like fingers. And we can also see that we get about 5 times normal enzyme expression in the bone marrow, and about 11% with the CD11B vector, which is actually quite a bit better than the original vector, so which is nice. Oops, there we go. The MPS IIIA mice are also very hyperactive. They love to run around. And you can see here that with the CD11B vector, where we have 11%, that we've got a correction of this hyperactivity. Whereas with the PGK vector, only 7%, we don't have a correction. And interestingly, there's actually a cutoff value. It's about 8.5% of normal levels in the brain. It's a little bit different in every disease, but in MPS IIIA, it's about 8.5%. That's all you need to get into the brain in terms of enzyme, at least in the mice. But we would hope it would be something similar in patients. And in fact, from metachromatic leukodystrophy, they got about 10% in the mouse model as well. So when they went into patients, of course, they've had a really pretty good effect with that level. So we're hoping to see something similar. We're also looking in the brain, and here are the brain cells in red, lysosomal, swollen lysosomes in green, and you can see the CD11B vector corrects this to normal levels. And the inflammation, which is brown, is also corrected right down to normal levels as well. So this is all good. In MPS IIIB, we've done something very similar, and to cut a long story short, exactly the same kind of mode of transplant. These things take absolutely forever, by the way, just in case you were thinking that we all did this in an afternoon. So it's about 10 years of work. But anyway, we see correction of hyperactive behavior. We see very high levels of NAGLU expression in the bone marrow, and compare it to a normal bone marrow transplant. Look at how poor that is in comparison. So that just tells you how poorly these cells are secreting enzyme. And we get about 13% of normal enzyme levels in the brain, which we hope will be enough again in MPS IIIB to get an effect. And again, normal transplant, WT-HSCT, so we use the term wild type to describe an unaffected mouse, WT. Don't ask me why we do this, but this is just tradition. And so WT-HSCT describes a normal transplant. LV-NAGLU is our lentiviral vector, and MPS IIIB and WT are obviously affected and unaffected, respectively. We also show reduction of the heparan sulfate in the brain, so MPS IIIB is very high, and of course with the vector we can see a full correction there. You can see actually there is a little bit of an effect with a normal bone marrow transplant, but it's just not enough. When we look in the brain, you can also see, so anything that's red and green is generally not good. There is a little bit of green staining here, red staining here in normal mice. This is because these are astrocyte cells, so you kind of require them. They have an immune surveillance role. They hang out in the brain, they look for trouble, basically. And in the MPS IIIB, you can see there's a lot of lysosomal storage material here in green and a lot of red cells in every region of the brain, which there shouldn't really happen. And you can see you've corrected this back to normal levels after the treatment. And a normal transplant is doing very little to change that. So for MPS II, we did something a little bit different. So we just published this. It's in this month's Embo Molecular Medicine. So what we did was that we took the IDS gene here. It's still being driven by the CD11B promoter. And what we did was that we changed the end. So we added on a little peptide, which allows the enzyme to cross the blood-brain barrier. So there are various transport systems across the blood-brain barrier because although most things are excluded from the brain, from the blood, there are some things you really need to get into the brain. So you need to get things like cytokines, you need to get some proteins into the brain, and those proteins that you need to get into the brain, they have an active transport system. So you can use a peptide which binds to one of these receptors and drags it across the blood-brain barrier. They're not usually desperately effective. Typically, if you did this with an enzyme, you might only get 1 or 2% improvement. So that's one of the reasons, I think, why it's typically not used that much in enzyme replacement therapy. But when you're overexpressing a product constantly, then it can actually make a big difference. So we figured it was worth a try. So same kind of protocol, bone marrow transplant, gene-modified bone marrow transplant. We're able to get more than 100 times normal enzyme levels in the periphery, so really, really high in the bone marrow. And again, we get a really high vector copy number, about 3-4 copies per cell. And we get full chimerism as well in the animals. All of these are important things. This is just showing that we've got a successful engraftment. And interestingly, we compared these 5 different treatments. So we've got a normal animal here, wild type, MPS 2 here, a normal transplant, WT-HSCT, the IDS lentiviral vector just without the peptide, and then the IDS ApoA2, which is the vector with the peptide, so with the blood-brain barrier crossing peptide. And what you'll notice is that this is an X-ray of a mouse, and the mouse has cheekbones. They've got a very nice cheekbone structure, as you can see here, very prominent cheekbones. But you'll notice that they're thickened in the MPS 2s. So you can kind of see that difference there with the arrow, hopefully. Can anyone see that? We also see the same thing. So these are called zygomatic arches. It's a very fancy word, but it just means cheekbones. And in the long bone of the arm, so the humerus, and the long bone of the leg, which is the femur, you can also see thickening there as well. And each of these treatments is able to correct that. So we were quite surprised that a normal bone marrow transplant was making a difference. So this is interesting, actually, because it means that the mouse is not exactly the same as patients, because I don't think we would see such a good effect on bones and joints in patients. We then look at 2 different outcomes. We look at the rotor rod, which is a spinning wheel that the mice have to run on. It gives an idea of coordination and balance. It gives a good indication of physical function. And then we look at the Y maze, which is spatial working memory, which is basically their ability to remember whether they've been to arm A, B, or C before. So essentially they go A, B, C, or whatever, something like that. But if they start going A, B, A, B, A, B, A, B, am I missing something? Shouldn't I go to C? If they start doing that, then basically that's a fault in working memory. So they've forgotten where they've been going before. So all of the treatments actually had a pretty good effect on the rotor rod, physical function, whereas on the brain-specific working memory, what we notice is that the MPS 2s don't do as well, but the only one that really is corrected back to normal is the IDS plus ApoE2 peptide. So we also looked in the brain. Again, just to be confusing, we've put them the other way around. So the neurons this time are in green. These are the brain cells. And the lysosomal material, the big swollen lysosomes, are in red. So you can see a lot of buildup in MPS 2. You can see that a wild-type transplant, normal transplant, it's almost exactly the same. So we're able to completely normalize the brain heparan sulfate levels right back to normal levels using the IDS ApoE2. And we get nothing with a normal bone marrow transplant at all. We're also able to change the sulfation patterning of heparan sulfate. So heparan sulfate is a complex molecule that allows cells to signal to each other. And the kind of biochemical structure of it is really important. So you can see that it's really abnormal. So here's in white, we've got a normal animal. Here we've got an MPS 2 animal. And you can see that we get a progressively better correction back to normal with the IDS ApoE2 peptide. So we wanted to know what was going on here. And it turned out that it wasn't just about crossing the blood-brain barrier, that actually through pure chance, total fluke, I have to say, that the ApoE2 peptide actually turned out to do some other things as well. And it was actually quite lucky for us that it did, because it's actually led to such an effective therapy. First thing that we noticed was that when we put IDS, or the IDS ApoE2 protein, onto brain endothelial cells. These are the cells which make up the blood-brain barrier. So this is a human brain endothelial cell there in a dish. We noticed that actually the IDS ApoE2 peptide protein was taken up much better, 3 or 4 times better by cells. And we thought, oh, that's weird. So we then did it in a hanging dish. So this is called a transwell. And what you do is that you're trying to mimic the blood-brain barrier. So if you imagine this is the bloodstream up here, and this is the brain down here, and you're trying to get the proteins across that barrier. So these cells are all sat down in a layer. They form a nice tight mesh. Nothing can really get through them very easily. And you're looking at the difference between IDS and IDS ApoE2. And you'll see that actually the amount that gets across this is really quite low. It's a relatively small percentage. But it is significantly better with the IDS ApoE2. So that is the blood-brain barrier crossing aspect. And then we really wanted to drill down to how exactly it was doing this. And the way that we do this is that we try adding in competing protein to block these receptors. So we either try and block the mannose-6-phosphate receptor, which, if you remember, is the normal receptor that takes up the enzyme, or we try and block ApoE-dependent receptors by using ApoE to block them. And to our rather surprise, it turned out that actually most of this uptake was being driven by mannose-6-phosphate and not by ApoE. And it turned out, you'll notice that we've added heparinase in here, that actually there was a binding site for heparan sulfate in the peptide sequence. And what this does is that it just makes the whole peptide much stickier. So it sticks to cells much more effectively. And you can knock that out by using heparinase. So when you knock out heparinase, you can knock out a lot of that binding as well. So it turns out that actually the ApoE2 peptide and the stem cell gene therapy using ApoE2 is not just working through increased blood-brain barrier crossing, it's working through increased cell uptake. So it's very similar to the GILT tag, for example, which actually works through increased uptake through the mannose-6-phosphate receptor. So we've then done quite a lot of preclinical workup for clinical trials as well. We got funded through the Valley of Death, which is a very fun part of developing work for clinical trials. You think you've done it all when you've done the mouse study. Actually, you're just at the very beginning. And what we wanted to do was to try and apply this program of work. We wanted to be able to take mobilized peripheral blood stem cells from patients. We wanted to be able to send these to a centralized GMP transduction facility, which is all very, sounds wonderful, doesn't it? So GMP is good manufacturing practice. This is the standard you have to adhere to if you're going to do these kind of studies. And we wanted to then freeze these cells so that we could evaluate them for safety and efficacy before they went back into patients. And to do that, we had to first make GMP vector, which we got funded by Great Ormond Street Hospital Charity. We then licensed the therapies to Orchard Therapeutics, so MPS 3A went to Orchard in 2016 and MPS 3B in 2017. But we continued working with Orchard on the preclinical workup. So we were really interested in doing full-scale transductions in patient cells so that we'd be able to take this to the regulator and say, great, we have a frozen product, we're going to put this into patients. And that's essentially what we did. We needed to show that our lab-grade vector was equivalent to our GMP vector, and we've done this in multiple different ways in human CD34-positive stem cells. We can show that we've got equivalent levels of SGSH enzyme. We can show that we've got a normal recovery of stem cell clones. So we plate the stem cell clones out on a dish. We expect to get the same number of colonies back, regardless of which treatment we do. If we don't get those colonies back, then it's because we have some toxicity, and we don't see that. So that's great. And then what we did was we did a full-scale transduction. So this is full-scale done at GMP. So it's all done in a clean room. It's an absolute nightmare. Luckily, Boston did this for us. And they sent us back the cells for us to evaluate, and we were able to achieve very high vector copy numbers in these cells. You can see from a single and a double transduction. And we were also able to freeze them and show that the freezing process didn't change that. So that was really nice. So we were able to show an effective scale-up in cryopreservation. Not content to wait there, we also needed to then do some work in mice, and these are immunodeficient mice. We needed to show that they would engraft just as well as cells which we hadn't transduced with the lentiviral vector. And to cut a long story short, they engraft equivalently. The immune cell subsets are all exactly identical, and we're able to achieve a really great engraftment in the mice. We also are able to show biodistribution in all organs except the gonads, which is, again, a really important regulatory finding. And we're able to show that despite the fact that these are normal cells, so you would expect them to express SGSH, we're still overexpressing SGSH in them. And I have to say, CD34 cells do not express a lot of enzyme, especially under the CD11b promoter. So there are quite a lot of clinical trials that have been run for genetic diseases. I'm just going to skip through most of this because they're all primary immunodeficiencies for the most part, with the exception of metachromatic leukodystrophy. But the point is that there's an approved drug out there for ADA SCID. So this stem cell gene therapy has now got an actual approval. So what are the trials that are out there for stem cell gene therapy in MPS? Well, there is actually an open trial recruiting for MPS 1 right now, which is in Milan. It's actually not with GSK, it's with the San Raffaele Institute. The MPS 2 work, we've only just completed the preclinical work, I'm afraid. So we still need to do GMP workup and take that forward for clinical trial. MPS 3A, we've done a lot of the preclinical workup, very close there, and Orchard Therapeutics will be taking that one forward to trial. And the same for MPS 3B. Orchard Therapeutics will be also taking that product forward to trial. And we think there might be some development in Mark Haskin's lab on MPS 7, but so far, actually, I don't know a lot about that. So, hopefully that was useful. Thank you very much.
Thanks very much, Brian. That's brilliant. Okay, so the next presenter is Dr. Harmatz, and presenting on managing the immunological obstacles in patients. Over to you, sir.
Thank you. It's a pleasure to be here. I should start by saying I'm not an immunologist or an allergist. I'm a gastroenterologist. So this topic can become very technical, but we'll try to keep it at the practical level that we deal with. And I could start by asking how many people have had an infusion-associated reaction here? Raise your hands just to, so maybe 20%, 10 to 20%. And how many did this stop you from getting infusions? Just out of anyone? That's good. That's the take-home message. If you learn nothing else from this, this talk, it's that we can almost always push through and find a way to give you the infusions in spite of reactions. So you've actually gotten the bottom line at the start. So these are my conflicts of interest. Today we'll talk a bit about immune reactions to enzyme replacement therapy, how we prepare for and treat acute reactions, some pre-medications that we would suggest, and even the extreme, which is desensitization for the patients that are really difficult to have difficult reactions that we're having trouble infusing. We'll talk a bit about the possible effect of immune response on efficacy, which is really it's a real challenge. It's a great, we know that has a great effect in Pompe disease and with Lumizyme and Myozyme, but it's been difficult to transfer the severity from Pompe to the less severe situation in MPS and to get a concerted therapy in this area. But I'll show you a couple of examples of immune modulation and tolerance that have emerged in the MPS grouping, and encourage people to, if you actually have this therapy, immune modulation or tolerance, make sure that your physicians report it, or work with a group that's trying to report a case series that will move the field forward. So Joe Munzer has talked to you about all of the progress we've seen over the last 10 to 15 years. We have multiple enzymes available for the different MPS diseases. They all are foreign proteins that are being infused in a patient that doesn't really know this protein. And for most of these enzymes, you'll see the prescribing information recommends antihistamine. This is empiric. It was used in the clinical trials. Whether it's having a huge effect, we can't really say, but it is listed with all of the enzymes except Elaprase. So you're probably used to seeing this with your infusions. Most people don't have much feel for what's happening during infusions. They haven't looked at what's called pharmacokinetic studies. And I'm showing you one set of data that we collected during the Naglazyme MPS 6 trial. And what you're seeing here are individual patients' infusions, and the top curves are one patient, and these patients got the standard dose that we use today, 1 milligram per kilogram. But the patients in the lower curve were at a 0.2 milligram per kilogram. So we didn't have a placebo in the trial, we just had a very low dose. And you can see that when we use what we thought was going to be the correct dose, we get a nice high concentration in the bloodstream, and we keep it up over 3 hours. And then as soon as we stop, in most patients, it disappeared very quickly. So this is one, it shows you our goal with these enzyme infusions. We really want to get above the sort of clearance of this material from the circulation. We want to see enzyme circulating, not being taken up by the liver. This way it can reach different organs. And some people wonder whether, can I do my infusion in 30 minutes, if I could tolerate it, if I didn't have reactions. This is one reason why we try to keep it up for some length of time, so it gives it a chance to move out of the bloodstream and reach different tissues. So it's possible you can shorten some, but I would be careful about really going to a very short infusion time. And Emil Kakis looked at this in his original dog model, and when he did very fast infusions, he got much less efficacy. So we try to maintain this infusion over a long time period. The same patients, we looked at the urine GAG in this graph and antibody development for individual patients. And you can see most patients had a very nice drop in the urine GAG, 60–70% drop. But there is this one patient that, with the open circles, that dropped and then went back up again. And you can see that this is the patient with the highest antibody that we had developed in the Naglazyme trials. So it was one of the first suggestions that most cases antibodies not affecting us a great deal with urine GAG, but it has the potential if you develop a really brisk response. This is the complexity of the immune system, and I'm not going to try to go through this in great detail. Basically, these are the antigens, the foreign protein, the enzyme that we give you each week. And when you first get it, it comes through and binds to certain cells. It stimulates the immune system. You get T and B cells that are sensitive. They make cells that make antibody against your enzyme. That antibody goes and binds to cells that then are triggered when they see the enzyme again, and they release the mediators that give you the reactions. So this is a complex system. There are drugs that affect different stages of the system, trying to, if you're having reactions, we can use those drugs to try to stop these reactions. But this is basically what is happening. You're seeing a foreign protein, or your children are seeing a foreign protein, and you're making an immune response. It's very similar to a peanut allergy or a bee sting allergy, penicillin allergy. This is the purview of the allergist immunologist, and one of my final recommendations will be for the very difficult reactions, you try to tie in allergy immunologist as a specialist to help you manage the reactions. So if you look at the prescribing information, you see very high numbers for antibody production. Elaprase has a little bit lower level. Keep in mind that this is completely dependent on the assay that was developed by the company. So if you have a very good assay, you'll detect antibody in everybody. And if your assay is not so good, you'll have lower levels. We don't really know what the levels mean. This is the percent of patients. Then you also get numbers, how much antibody do you have. So if you have a huge amount of antibody, you have a high titer, you can dilute it and dilute it and dilute it and still have antibody detected. So there's a lot of information that's not present in this type of number. The same is true with these other numbers. Neutralizing antibody depends, it's in the literature, it really depends on the type of assay, the quality of the assay, whether this is present for 1 or 2 samples, but not the whole course of the trial or at the end of the trial. And by and large, most of the companies have come to the conclusion that overall they don't see that these antibodies are related to efficacy. And I caveat that we've shown that very high titer may affect some efficacy, and also they don't think they're related to immune reactions, although when Shire looked very closely in one study, they find that the patients with a much higher titer do have a higher likelihood of having hypersensitivity reactions. So overall, antibody is not terrible. Everybody wonders why people are not measuring it. We don't really know how to it doesn't affect us or make us change therapy, so we tend not to follow it very closely. But we may look at it more carefully if we're having problems with reactions. So going back to Emil Kakkis, he opened the meeting. He's one of our main mentors, and when he first started working with the dog model of MPS 1, he was giving enzyme, it was foreign, it was human, to the dog model, and he was getting tremendous reactions, and he had to get over that problem or he wasn't going to be able to study enzyme therapy for the MPS 1 dog. And what he discovered is that if he could give the infusion really slowly for an hour, it seemed to consume the reactants. So everything was used up in that first hour, and then he could speed up his infusion and give it successfully. He also added an antihistamine to the mix, but I think he really feels the slow reaction part is really the critical part. And what he noticed that there's this material called complement that is part of your immune system, and when you stimulate during that first reaction period, you're releasing complement, it's being broken down, and that's giving you the side effects, the anaphylaxis, the drop in blood pressure. So by going very slowly, he was able to overcome that problem. And that's one reason for Naglazyme and the MPS 6, we used a protocol where we gave 3% of the infusion in the first hour, and then 30%, 30%, 30%, and that finished the whole infusion. But you can see how slow that first hour is. And, I encourage people, unless your physicians think they have a different way of overcoming that early phase by sort of cycling up the infusion over an hour, some other method, that this is probably a critical part of giving this medication. We have definitions that are confusing. We have anaphylaxis, we have anaphylactoid reactions, we have hypersensitivity, infusion-related reactions, infusion-associated reactions. These are really definitions that won't mean much for you, but you'll read them in the literature, and the FDA uses them a lot, and you have to categorize reactions to fit these different categories. The one that probably is most relevant is just to consider them anaphylactic if they're the typical reactions of urticaria, GI symptoms, vomiting, blood pressure drop, respiratory distress, any of those together, and usually FDA wants to see two different body systems to define it as an anaphylactoid reaction as the most important category. The biggest difference, anaphylaxis just means that we know it's related to one type of antibody called an IgE antibody. If it's not IgE, then it falls into anaphylactoid. Hypersensitivity is a much broader, it includes other types of immune reactions and infusion-associated reactions. When you're doing your clinical trials, they would like to have any reaction that happens during an infusion reported as an IAR so they can track that as a problem. Christine Haller put together this description with Emil Kakis's help. It was reported during the MPS 7 trials, and they really divided the reactions that we should be alert to into four groups: the anaphylactoid, which is most of our infusion reactions; anaphylaxis, which is IgE mediated; urticaria; and then immune complex reactions. So it helps us to categorize and group because some of the symptoms fall into one group. The immune complex don't have to happen during the reaction. There you have your enzyme, you have antibody, you form complexes, you deposit the complexes between those two materials in different tissues, and you can have that reaction later, a day later, 2 days later. It can cause some kidney effects, can cause fever, it can cause muscle joint aching. So it's quite different and it's rare. Urticaria we see a fair amount. Anaphylaxis is hard to define because we don't measure IgE, and most reactions are anaphylactoid. They occur early in the infusion period. They can involve mostly respiratory, but you can also get blood pressure. You can get some cramping GI symptoms that fall into this group. How frequent are these reactions? Again, this came from the prescribing information. If you go back, if you want to learn about your specific drug, look up online. You'll see very detailed information that's prepared with FDA called prescribing information, and it goes through all the information in summary that the pharmaceutical company submits to get approval for the drug, and so you can learn a lot. But it's hard, again, the numbers sometimes don't mean a lot. They have a frequency of reactions, 49% of the patients for MPS 1 in the clinical trials, but that doesn't tell you how many reactions. So this patient is counted as positive, and may have had 1 or 2 or 3 infusions with reactions, but over the course of the year, got 50 infusions, and in 2 years, 100 infusions, and had 2 reactions. So it's deceptive looking just at a percentage and trying to judge, do we have more reactions to ylsulfase or galsulfase? It's very hard to compare from the different patient populations, number of patients, different ways of counting these reactions. The thing that is clear is most of these reactions are mild or to moderate, that we very rarely get severe reactions. So you have to be prepared, and we'll keep coming back to that. If you're doing home infusions, be prepared. If you're in an infusion center, they need to be prepared. I'll show you an example of the only time I had to give epinephrine in 20 years of infusions and thousands of infusions. So you can see how rare that is. But when I'm traveling and I present discussions of immune reactions, there's always one person that has had very difficult reactions and hard to manage reactions, so you have to be ready for it. So most are in this category. We get estimate as 1% of patients can have a severe reaction. I probably have had an even lower percentage. And then very rare you have this immune complex mediated type of reaction. Symptoms are quite similar with the different drugs. I wouldn't try to associate one specific symptom with a specific drug. This is called black box warnings. The FDA labels, they will work with the companies to design the label and write the entire label, and sometimes they will attach what's called a special warning, and virtually all the enzymes have a warning about anaphylaxis. Naglazyme has a warning about cervical spine instability, and within they don't have a clear black box, but they have warning about anaphylaxis with Naglazyme. So it really is a foreign protein in all cases and can potentially have the same reactions. These are some of the sets of equipment that you should really have available if you're infusing, even home infusion. They should have much of this available to deal with reactions. We have some protocols. We have different nurses working with these patients, especially within the clinical trials. We have to have pretty set protocols and we have one for mild reaction, we have one for moderate and then severe reactions. And the most important thing, the first thing to think about if you're starting to have a reaction is either slow or stop the infusion. Your nurses have to really be trained. That's the knee-jerk reaction. Don't try to page somebody or discuss it with pharmacy and leave the infusion running. So just stopping the infusion will stop most of the mild to moderate reactions. So miscellaneous recommendations with infusions. We try to avoid giving infusions with an acute illness. The patient has a fever already. It's hard to recognize a reaction. We wonder if we may actually set people up. The immune system's being modulated by the infection, so you might be more susceptible to developing an infusion reaction or sensitizing to the drug, breaking down your tolerance for the drug. All of those are theoretical problems, but we respect it if someone is just starting an illness. We warn people if you have sleep apnea and you fall asleep during the infusions, you should have your CPAP there. You could have apnea and it could be a complication of the infusion, but unrelated to the drug. That same caveat applies to if you're using sedating antihistamines. Be careful. If you fall asleep with a sedating antihistamine, the same thing could happen if you have sleep apnea and you're not using your CPAP. We generally pre-med most our infusions. We don't for Elaprase. It's not in the labeling. It wasn't used in the trial. Another very theoretical issue is whether to vaccinate on the day of infusion. And there is one anecdotal case with Vimizim where in an early phase, patient received a vaccination the same day and the next week had a very severe reaction. So it makes sense to me not to give another foreign protein the same day with an adjuvant where I'm hoping for immune response same day as an infusion. I'd rather let the infusion clear for a couple of days and then move to the vaccination. We also warn people not to shake the drug, prepare it very gently. Aggregates tend to sensitize more and you'll aggregate the protein if you're really rough with it. So be careful during preparation. Warn your pharmacist or your home nurse if they're preparing it at home not to create bubbles or shake it or disturb the protein. So this is an example. If we're really having trouble, probably the most helpful maneuver is not changing your pre-meds or adding steroids the day before. It's really slowing the infusion down to the point where we call it desensitization, and we'll drop the concentration way back, even lower than that 3% in 1 hour, and we'll gradually build up the amount of drug. And you may find you don't go to the full infusion, you're only going to give 0.5 milligram instead of 1 milligram per kilo, and you stop early depending on how much time you have available. But there are examples in the literature of desensitization and being able to do it for a time period and then move back to standard infusions. Patty Dixon's given us some animal data looking at the effect of anti-drug antibody. We have a little bit of human data to look at. I showed you one example of a urine gag that was modified. This was the phase 3 of the Naglazyme trials. You can see the patients, this is the amount of antibody they have, and this is their urine gag. And when you get to look at the patients that have the most antibody, you see that over time they seem to have worsening urine gag. Most of them went up in urine gag as the antibody titer or amount of antibody went up. Same was reported by Ed Wraith in the MPS 1 under 5 study. You can see here, this is urinary gag, this is antibody. So when the antibody amount is lower, the percent reduction in the urine gag is higher. When the amount of antibody moves up, the amount of reduction in urine gag is lower. So again, another effect of antibody on the ability to respond to your therapy. Here's examples from Patty Dixon's laboratory. She was able in the dog model of MPS 1 to induce tolerance, and she found that the actual tissue levels of gag were much lower when she induced tolerance as opposed to a non-tolerized animal. So this is the tissue gag. This is tissue gag in tolerant animals, and she had a 45% reduction. When she gives an antibody and then the enzyme, she found that the distribution of enzyme was different, and in fact, it went much more to sinusoidal cells in the liver, for instance, instead of the hepatocytes where you'd really like it to go. So antibody, even though we get by without reactions, we're probably having a modification in how responsive we are. How do we take care of that antibody? Do we have any way of changing it? We don't really right now. It's just really dependent on each individual patient. But we're beginning to get examples in the literature of people that are actually trying to very actively modify it. And the first example was from Catherine Kim and Barbara Burton's group, and they had described here a patient. Both of these are MPS 2 patients. This patient had a very severe large deletion. A 6-year-old had been started on enzyme therapy, had no drop in urine gag, so she was quite frustrated. And she brought in Priya Kishnani's protocol for modulating antibodies. So this patient had a lot of antibody, was very sensitive, but you can give a series of drugs that will knock out the antibody, and it'll stay low for a certain time period, then you have to repeat the series generally. But what she did this procedure, was able to get some reduction in urine gag, some partial improvement in clinical symptoms. So this was and she had to treat pretty aggressively with Priya's protocol, but the patient did have a response. The more exciting study is the one from Kara Wolgar. Is Kara here? Here's Kara. Her poster was up yesterday. She should be up on the platform. This is a pretty remarkable study. It's a 3.5-year-old female diagnosed with MPS 2. She has 100% exon activation and an inversion, a large inversion, and was once they diagnosed her, they predicted she would be one that would develop a lot of antibody. And so what they did was immune tolerance induction. You have to start the enzyme at the same time as the Priya Kishnani protocol for modulating antibody. And then she treated for a month 4 doses of rituximab to modulate the B cells. She used methotrexate. And a year after completing it, the patient now is not receiving any immune modulation, has normal IgG, is negative for anti-drug antibody, and has gone from a 5-fold elevation above normal urine GAG down to normal urine GAG. So here's a very successful modulation in an appropriate patient, and if we can get 10 of these cases into a series, it'll make a huge impact. That's how Priya was able to modify treatment for Pompe disease, severe Pompe disease, and hopefully this will be the mechanism in these patients. So this is a case that's reported by Barbara Burton. It was the first MPS 6 patient receiving commercial infusions. They had after 3 or 4 infusions, started having severe reactions, and the most important thing they did was lengthen the infusions to 16 hours. It took a lot of infusion center, hospital care, but by a year the patient was back to a normal protocol and is now on home infusion. And you just have to be persistent, you have to not give up with the process. And then I'm going to finish with R1 severe infusion reaction. It was iatrogenic. It just shows you that even if you're doing home infusion, experienced nurse, you need to be prepared for the major reaction. And this was a patient that was had been tolerating infusions, 10-year-old, was almost 6 months into a protocol, and infusion was started at the low rate. It's really low. Sometimes when you have a smaller child and a small volume bag, you're infusing not 3 mils per hour, but 1.5 mils per hour. And they had this is the standard tubing that's close to the patient. So this is where the patient's IV is. This is the line leading into it. And blood was backing up in the line. So the nurse had normal saline, thought, okay, I'm going to protect the filter. So she pushed normal saline in. But what you have to remember is you have full concentration drug, and even though the volume's very small, you're really pushing a significant amount of drug. And within 2 or 3, 5 minutes, the patient looked dusky, pulse ox was dropping, and we had to move quickly. As soon as we administered epinephrine, the symptoms reversed, and the patient did fine. We stopped the infusion for half an hour, gave some extra meds, and then restarted the infusion. So this just shows you that you can have reactions that are unrelated to a mechanical problem or an iatrogenic problem, or you have to be prepared for the iatrogenic problem. The other typical time this happens is when you're right on the border of going between a 100 mil bag and a 250 mil bag, and people, they're going back and forth because your weight's varying, and you actually have an order for a 250 bag, and it's put into a 100 bag, and you run faster, and you can have a reaction at that point. So be prepared. That's the most important thing. So I will finish there. Thank you for listening. Encourage you never to give up on giving the infusions. We can almost always find a way around it. Call Joe Munzer if you have any problems. He's sitting in the audience, and we'll be very happy to guide you through these infusions. So thank you.
Thank you very much, Dr. Harmatz. Okay, I appreciate we're running somewhat over, so I'm going to immediately hand straight over to Terry Klein to moderate the next session, and we're going to carry straight through.